I want to begin this seminar by arguing that the time is, it's a Kantian sort of argument that time is a fundamental aspect of experience, and that it is also primitive. It's fundamental because the experience of time appears to, or the representation of the time at which an event occurred appears to be fundamental, essential to our ability to recall that event. The argument for this comes from the phenomenon of retrograde amnesia, which I'll give a brief account of later in the talk. And secondly, time, as I've argued with my frequent collaborator, Peter Balsam, whom I'm very pleased to see in the audience, is a foundation of associative learning. Associative is in scare quotes because both Peter and I are somewhat skeptical about the idea that there is such a thing as an association, as traditionally understood, that what's really going on in associative learning is the animal is learning the times, the intervals that elapse between the events that it experiences. But whether one believes in associations or not, there's little doubt that this ability to anticipate what's coming is one of the fundamental elements of cognition. And for that, you have to represent time. Secondly, the claim is that it's primitive. I mean this in the evolutionary sense, because memory, as I will show it early on, almost immediately here, memory for time, that is where in the day, in the 24-hour cycle, things occur, and memory for the duration of intervals are observed even in insects. I thought for an interdisciplinary audience that might be news. And they're also observed, as I'll show later in the representation of time, is observed in decerebrate vertebrates. Those are vertebrates. The decerebration is removing the entire front 3/4 or 4/5 of the brain, all the parts that neuroscientists ordinarily talk about, and leaving only the parts of the brain that appeared, that are seen in the earliest and most primitive of vertebrates, the hindbrain and the midbrain. But even, as I'll show you experimental results, not from my laboratory, showing the representation of duration in the cerebellum, which is a kind of the cortex of the midbrain. So I think the fact that we see these in animals, a species as diverse from ourselves as insects, and in these very old parts of the brain, suggests that the basic mechanisms for measuring and recording time evolved long ago, in probably in the Cambrian or Precambrian, and that suggests that they were already present in the individual cells before the emergence of multicellular organisms. Now, there are two aspects of time. There's time itself, which, can I point to things here? Yes. Okay, so, oops. So time itself is a point. When you say, what is the time at which something happens? Well, I said 10 o'clock. Well, what is 10 o'clock? Well, 10 o'clock is a point on a circle, right? It's, more technically, it's a phase in a phase point in a physical process, cyclical physical process, and we can damn it. Yeah, maybe a pointer will.
It's right
There we go. Thank you.
I feel that way. Yep,
Okay.
There you go.
Yes, okay. So, one way of representing cyclical processes of any kind is in what's called a state space. A state space is defined by at least two variables. In the case of cyclical processes in individual neurons, these are biochemical variables within the cell. And as one variable increases, the other may decrease, and then this variable remains high while this variable increases, and then as you go around and around in state space, and at any point in state space is defined by the combined values of those two variables, and that defines a phase. And then the duration is the difference between two phase points. That is the phase angle in this circle. Durations can also be measured by decay processes, as in C14 dating, and there are many neurobiological theories about the representation of time that postulate decay processes, but I think the evidence in general favors the idea that the physical basis for the brain's ability to represent time derives from these cyclical processes that exist even in individual neurons. Be that as it may, I promised to show you that even insects are aware of the time at which things happen and make surprisingly sophisticated use of their memory for the time of day at which something happens. In this experiment, which is a very old one run by Wall back in the 30s, he was a student of von Frisch, the discoverer of the bee dance. There we go. So he had, here we have the diagram of the, so he had, these were done with honeybee subjects. Here was the hive, here was a feeding station, here was another feeding station. Wall stocked the beaker on this feeding table on a series of days prior to the day on which these data were generated. He stocked this feeding station between, what do I say here, 1530 and 1700, that's the gray here, each day. So during that part of the day, there was no sugar water here, but there was sugar water here, right? And in the morning at station B, he stocked this station with sugar water between 9 and 10:30. So in the morning, there was nectar, artificial nectar here, and in the afternoon, there was nectar there. And these data were generated on a probe day in which there was no nectar at either table, and there was just an observer standing there noting how many bees arrived and what they did. And what you see here is that in the morning, that is, in anticipation, notice that the peak arrival time for bees in the morning is just before the onset of the expected presence of nectar at this. By the way, to eliminate some of the mystery, I should explain that different species of flowers produce nectar at different times of day. So this has an obvious adaptive function. That is, the bees learn when the different flowers produce nectar during the day. But you see, the thing that interests me most about this is that I'm plotting total visits here, and the black indicates the proportion of those visits in which the bee actually landed and went into the beaker to make sure whether there was or was not nectar. So you see that during the morning, the bees had a much higher expectation that there really would be nectar here at station A than they did at station B. They went to keep in mind, when they went to station A, they didn't find anything, right? So many of the bees then went on to station B, right? But they clearly had a stronger expectation of nectar at station B in the morning than they did at station A in the morning, as evidenced by the fact that they actually landed and went inside. Now you might ask, why do they go check out the other station without landing? Well, because bees use the presence of other bees to indicate whether something good is going on. So a flyby at the other station to see whether there's anything doing there is well worth it. And this pattern reverses in the afternoon, right? So now, notice, by the way, that they don't come to either station during the periods when there was no nectar on the training days. And now in the afternoon, the much greater proportion of the visits to the A station are serious visits. Actually go in and look for yourself, as opposed to flybys. So the bees evidently have learned the 2 different times of day at which nectar is available at the 2 different locations and are making rather sophisticated behavioral use of that information. So I'm trying to argue here that these representations of both phase points, which is what I was just showing here, that is time itself, and duration, which is what's shown in this slide, are present even in animals that we're inclined to look down on. And the mouse perhaps isn't quite as lowly in most of our estimates as the bee, but it still is often looked down on. Now this shows data that I'll be explaining here in which, from an individual mouse, in which it's representing 3 different durations. So in this experiment, the animal's in a test box like this, and when when a signal came on, the animal could get fed after a delay of 5 seconds if it went to this hopper, which I've called black only because it's graphed with black over here in the figure. It wasn't actually black. During one part of the experiment, this part of the experiment, but he didn't always get reward for going there, right? So the signal would come on and he would go here, and sometimes he'd get his reward at the end of 5 seconds and sometimes he wouldn't. And what you see here, this is a plot of the time elapsed in the trial. It's on a log scale, but it's, what you see here is that a whole sequence of trials. As you go from top to bottom, you're going through a sequence of trials here. And you see that when this was the only place where food was to be expected, the mouse's visits to this location after the signal came on bracketed the expected time of feeding at that. So he came looking for the food, and if it arrived, he ate it, and if it didn't arrive, these data come only from the, I forgot to mention, these data come only for the trials when the food doesn't arrive, okay, the so-called peak trials. So the animal comes, he waits for his food, he doesn't get it, he no doubt mutters some imprecation and leaves, right? So the same subject later on, another block of trials, the reward was delivered after a longer delay, namely 15 seconds, and only in this hopper. And again, sometimes yes, sometimes no, and those data are plotted here. So you see now the animal tends to arrive later, and but his visit brackets the expected time for food delivery in that hopper. And then here's a series of trials where the delay is in this hopper, and it's 45 seconds, similar story. And then finally, the most interesting part is the final part of the experiment. When the signal goes on, all 3 of these hoppers light up. And 1/3 of the time, there's a reward available after 5 seconds in this hopper. 1/3 of the time after 15 seconds in this hopper, and the final 1/3 of the time after 45 seconds in the last hopper. But the mouse has no way of knowing which hopper is actually going to pay off. So what would you do? Well, I would go first to the 5-second hopper, and if I didn't get my reward, I would then go to the 15-second hopper, and if I still didn't get my reward, I would go to the 45-second hopper. And as you see, that's exactly what the mouse does. And this tells us how precisely the mouse can actually represent the time, right? Which, notice, is much tighter than you would have thought from this behavior here, right? When we sandwich the 15 seconds between 5 and 45, we get very tightly timed behavior. So this mouse has learned the 3 different durations. Now, the jumping levels of analysis from the behavioral level now to the what's my time doing here? Yeah, I'm doing okay, I think. To an incredibly exciting experiment that was published from the laboratory of Jerry Heslow in Lund, Sweden, published in 2014. And they're recording from individual Purkinje cells in the cerebellum of decerebrate ferrets. I mentioned earlier what decerebration is, right? The importance of that here is that we know, because we've removed the hindbrain and the midbrain from all the rest, from the entire forebrain, we know that everything we're looking at here depends only on what's going on in this very primitive part of the brain. And they did with these individual neurons an artificial experiment that's exactly like Pavlov's famous experiments with the dog, where he would ring the bell, which we conventionally call the conditioned stimulus, and after some delay, he would deliver food to the dog. And after a number of trials of that kind, the dog would begin to salivate in response to the ringing of the bell. What is often not mentioned is that the dog doesn't begin to salivate just at as soon as the bell begins. There's a 30-second delay between the onset of the ringing and the delivery of the food, the dog begins to salivate after about 15 seconds. If there's a 30-minute delay, the dog begins to salivate after about 15 minutes, and so on. So in other words, the dog has learned the duration of the latency between the onset of the bell and the delivery of the food. The Johansson et al. Experiment from the Hessel lab is doing exactly that experiment, except there's no bell, there's no food, there's just direct electrical activation of elements, individual elements of the nervous system. Instead of ringing a bell, they are directly stimulating the parallel fibers, which are a massive input to these cerebellar neurons. I'll show you a schematic here in a moment, but I want to make sure you get the gist of what's going on here. So the Purkinje cell is this huge neuron with this enormous dendritic tree, and there are 200,000 or more parallel fibers that come and synapse on the dendritic tree. And they are directly electrically stimulating the parallel fibers that are the input to the Purkinje cell. And that's functioning as the CS, the equivalent of the bell in Pavlov's experiment. Each Purkinje cell, so it gets 200,000 parallel fibers, it gets one climbing fiber. And they're directly stimulating the climbing fiber as the equivalent of the food. So this cell is getting two kinds of inputs: the parallel fibers, which are being artificially stimulated to serve as the CS, and the climbing fiber, which is being artificially stimulated to serve as the US. And there's a fixed latency between the onset of the stimulation of the parallel fibers and the onset of the stimulation of the climbing fiber. That's the same as the latency between the onset of the bell and the delivery of the food in Pavlov's experiment. And what the experiment shows is that this individual neuron is learning and remembering and reproducing that latency, the latency between the onset of the CS and the onset of the US. So here's the diagram that I promised. Here's this enormous Purkinje cell. It's one of the largest cells in the brain with this huge arbor here. The parallel fibers run like this. In this experiment, they have what they call the CS electrode, which is exciting these parallel fibers. Here's the climbing fiber, the other input, and here's the US electrode stimulating the climbing fiber. And the Purkinje cell normally fires at an extremely high rate. It's a spontaneous firing rate. It's not driven by any input. Often fires around 80 hertz, 80 spikes a second. And when they first begin training this individual neuron by turning on a 800 millisecond train of stimulation to the parallel fibers, followed after, now, I call these preparations. In one preparation, one thing, they may follow the US stimulation follows the CS stimulation 150 milliseconds after the onset of the CS stimulation. Okay? But the CS stimulation in this particular experiment actually continues for some while after the delivery of the US stimulation. In another version, the next day, when they're working with a different Purkinje cell, instead of using 150 milliseconds, they use 300 milliseconds. And on yet another day, with yet another Purkinje cell, they might use 450 or 500 millisecond delays between the onset of the CS stimulation and the onset of the US stimulation. They can actually sometimes, if they hold the cell for a really long time, train the same cell with two different delays. First they train it with one delay, and then they train it with another delay. So what you would expect from conventional electrophysiology is that when you turn on the stimulation, the rate of the Purkinje cells should, if anything, go up, because the parallel fiber input is glutamatergic, it's excitatory. And indeed, that's what happens initially. But after some repetition of trials, this cell no longer increases when the CS comes on. It shows a profound inhibition, a profound pause in its firing rate, and that pause lasts an amount that is closely proportional to the interval, the latency with which it has been trained. So it stops firing when the CS comes on, even though it's being driven by this CS input, and it resumes firing at approximately the time at which the US stimulation is expected. So that's what I've described here. And this is true on probe trials where you don't deliver any US stimulation, right? You just, you ring the bell but no food. So you stimulate the parallel fibers. Again, you see this pause. And the pause, the most astonishing aspect of this experiment is that the pause is independent of the parameters of the input stimulation once the cell has been trained. So you can make the CS input much shorter, much higher frequency, much longer. It doesn't matter. You always get the same pause, and the duration of that pause is the duration on which the cell has been trained. So, the point is that the response of the cell depends on what it remembers about the past, not upon the patterning of the current presynaptic input to the cell. I'm running out of time, so I want to make sure you understand how startling this is. Just a schematic of what's going on here. Here's our CS electrode. Here's where we're recording. What lies between where we're stimulating and where we're recording? Well, only the monosynaptic input. Yeah. And so it can't be in the synapse, so it must be inside the cell itself. So very quickly, on retrograde amnesia. Retrograde amnesia is the most common of all neuropsychological problems. Whenever people get a bang on the head, if you test them carefully, you'll see some degree of retrograde amnesia. There are two fundamental facts about retrograde amnesia you need to know. The first is that memories are inaccessible entirely based on the time at which they were acquired, independent of how important or trivial they are. That's the first thing, so that the time at which they are acquired makes the memory inaccessible. The second thing is that it is a problem in accessibility, not storage. Because the second important fact about retrograde amnesia is the memories almost always come back eventually. And again, they come back on the basis of the time at which they were laid down. So the person comes in with a concussion after an automobile accident. They don't remember the fact that they were married 20 minutes before they hit the tree, but they do remember rehearsing for the wedding a week ago. And as they spend days in the hospital, the memory up to the time of the wedding begins to return. So, in conclusion, A subjective time is a point in the state space of a biochemical cycle in one or more neurons. That is the phase point that I showed you. Subjective duration is a difference in phase points, a biochemical phase angle. Mechanisms for recording phases and phase angles evolved very early and are very widely dispersed in the animal kingdom. They appear to be properties of individual cells, and they are foundations of cognition because, as Kant realized, you can't deduce time from experience. The representation of time is a precondition for having an experience.